Now let's take a look at the eukaryotic cells. And so here in this overview slide, you have sort of a half animal cell, half plant cell. So the bottom part here is animal, which is has no cell wall, and then up here in the plants you see green, the chloroplasts for photosynthesis. Obviously the big difference between eukaryotic and prokaryotic cells is that nucleus, and so here the nucleus is big fat in the center right there. We also have a nucleolus here, and then we'll talk more about these other structures that are found in the nucleus. In addition, you have all of these endomembranes, this endomembrane system in a eukaryotic cell that you just don't have in a prokaryotic cell. So the ER, the endoplasmic reticulum, and then the GOlgi apparatus, all of these little vesicles, none of that you would find in a prokaryotic cell. But in eukaryotic cells, you do have that. Then the cell wall of plant cells, it's made of cellulose, and then the animal cells, they don't even have cell walls. They may have cell extensions, and here you have a long flagellum. Now that is something that you also see in the prokaryotic cells. Here you have a transmission electron micrograph of a plant cell, and of course the typical thing for plant cell, that would be the chloroplast, and then out here the cell walls. A lot of plant cells also have vacuoles for storage purposes. Okay, moving on. So let's take a look at cilia and flagella. These are projections used for movement, and the flagella are very long. The cilia are short projections. They are different in structure, but other than that, they have some resemblance to the flagella, for example, of a prokaryotic cell. So here, let's take a look at this flagellum, the picture right here, very long kind of extension here, and then the cilia very short, hair-like projections of the cell, and that's an electron micrograph. So here, let's take a look at flagella and cilia, sort of what they're made up of. They consist of microtubules. They are made of the protein tubulin in an alternating fashion, and these microtubules, they are organized in 9 pairs in a ring plus 2. So 9 plus 2 array, that's what we call that. That's the typical eukaryotic flagella and cilia arrangement. And then these flagella, they move in this wave-like manner to propel the cell. So here be this typical 9 plus 2 arrangement. You can see 9 pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. These are duplex or double microtubules. And then we have the central microtubules. There's another 2, 1, 2. And that's the 9 plus 2 arrangement of a eukaryotic flagellum or the cilia. And now let's take a look at the cell wall and the glycocalyx. In prokaryotes, just to sort of recap that, we have the peptidoglycan in the eubacteria, that is. And here in these eukaryotes, we find a variety of cell wall materials. Now, cell walls are only found in plants, fungi, and algae. So no, not in animal cells, and not in animal-like protista even. So cell walls only found in plants, and then algae are sort of the attachment, single-celled plant-like protista, and then the fungi. Okay, the cell wall is made of carbohydrates, various types of carbohydrates, various arrangements of these sugars, and how they're bonded with one another. For plants, we have cellulose. It's a material that you should know. For fungi, we have chitin. Chitin is kind of an interesting material. It's also the exoskeleton of bugs, and so if you step on a bug and it's got this crunchy kind of sound to it when the chitin breaks, yeah, so that's chitin exoskeleton. And then in yeast we have glucan, mannan. So, but the important ones here for plants, cellulose and chitin. for the fungi. The glycocalyx, those are also carbohydrates. They're bonded to proteins and lipids in the plasma membrane. They would be found in animal cells, and they help you, a complex eukaryotic organism such as a human being, to help the immune system to recognize self, distinguish it from non-self. So the glycocalyx has a variety of functions. It also bonds the cells together and helps tissues to sort of to stay in place. And here, take a look at the plasma membrane. The plasma membrane, similar in structure to the prokaryotic cell membrane in that we have phospholipid bilayers, and then we have the typical integral and peripheral proteins. Integral meaning that they span in the entire phospholipid bilayer. Peripheral, they are sort of anchored but not embedded in the phospholipid bilayer. There's some differences here in structure. The sterols, so like cholesterol, that would be only found in the animal phospholipid bilayers embedded in it. And then we also have these carbohydrates for attachment and cell-to-cell recognition. So let's take a look, closer look here at the plasma membrane. The function is similar between prokaryotic and eukaryotic plasma membranes. We have selective permeability. That means we have channels and different pores that are embedded into the plasma membrane, which are to help the organism to facilitate diffusion of substances that normally could not cross the phospholipid bilayer. And so, of course, we have also the movement of water down a concentration gradient, which we know as osmosis. And then active transport, those will be transport molecules such as ion pumps that will transport stuff actively, and they can be also embedded in the plasma membrane. Now, some differences in function here, namely endocytosis, and phagocytosis. Those are things that you find only in the in animal cells or in eukaryotic cells. So endocytosis means we take something in. That means we form a little vesicle and then we internalize what we wanted to take in. If it's something solid that we are engulfing and internalizing, then we call it phagocytosis. That term is known. Phago means to eat, and cytos, cell. So the cell is eating something solid. If it's something liquid that the cell takes in, it would be penocytosis. So penocytosis means liquid eating, or we're drinking something. And so in the phagocytosis, there's these pseudopods that are extending and engulfing particles. And in penocytosis, the membrane folds inward and brings the fluid substances inside of the cell. In the cytoplasm, so the cytoplasm defined as the substance inside the plasma and outside the nucleus, and there we have the fluid portion of the cytoplasm is called the cytosol, and then the cytoskeleton is sort of the infrastructure of the cell, and it's made of these microfilaments, intermediate filaments, and then even microtubules, which are larger than intermediate filaments. And they work just like your skeleton. They give the cell shape and sort of infrastructure support. We also have cytoplasmic streaming. That's the movement of cytoplasm throughout the cell. And now here's a big one, the ribosomes. This is something that you must know. The 80S ribosomes, those are for eukaryotes. And then we have 70S ribosomes. Those are the ones for prokaryotes. That difference is huge. You may not think of it that way, but it is very big, because that is the basis of how many antibiotics work. They bind to the 70S ribosome of the prokaryote, but not the 80S ribosome of the eukaryote. So the eukaryotic ribosome consists of the large subunit, the 60S subunit, and the small subunit, 40S subunit together, 60 plus 40. This is biology math. I have to tell you, 60 plus 40 makes an 80S particle, but that's just because it's more dense when you put the two pieces together. Many of the ribosomes in eukaryotes, they are membrane bound, so that means they're attached to the endoplasmic reticulum, making it the rough ER that you see in eukaryotic cells. We also have some free ones, like in prokaryotic cells, they're all free, and so they would be in the cytoplasm. Now, in the prokaryotes, we have the 70S ribosomes. They are composed of the 50S small subunit and a 30S small, the 50S large subunit and the 30S small subunit, and together they make a 70S particle. So that is the ribosome that prokaryotes have, and in eukaryotes, it's interesting, we find them also in chloroplasts and in mitochondria, and that's where this endosymbiotic theory comes from. The chloroplasts and mitochondria of the eukaryotes they were originally prokaryotic cells that took hold in eukaryotic cells. We can talk some more about that. But anyway, be sure that you know the difference in the ribosomes between prokaryotic and eukaryotic cells. 80S for the eukaryotes and 70S for the prokaryotes. Okay, so here I just want to point out the second part here. The antibiotic erythromycin binds with the 50S portion of a ribosome. 50S, remember, that would be the large subunit of the prokaryotic ribosome. What effect does this have on a prokaryotic cell? Well, it will hinder protein synthesis, and when something is bound to this portion of the ribosome, then protein synthesis can no longer take place, and so the cell will not be able to grow and multiply, and so therefore we are limiting the growth of a prokaryotic cell. Now, on a eukaryotic cell, we don't have a 50S large portion of ribosome. Remember, here, let me go back. In a eukaryotic cell, we have a 60S subunit and a 40S. So we don't have the 50S that the erythromycin binds to, and so therefore we have a nice drug that limits the growth of prokaryotic cells without harming the eukaryotic cells, and so we call that selective toxicity. It's only toxic to certain cells but not others, and that's a great thing. Now let's take a look at the nucleus, and there we have a double membrane bound organelle. This double membrane, we call that the nuclear envelope, and it contains the cell's DNA and a significant amount of protein, which is mostly these histone proteins. Together, DNA plus protein forms what we call chromatin. And now that's nice that we have the nucleus and that we protect our DNA. It does present a bit of a problem when you want to divide, because there's only one nucleus, and both of the new cells after mitosis, after cell division, they need to have a new nucleus. So during mitosis, meiosis, the chromatin needs to condense, and it's a fairly complicated procedure to do cell division in eukaryotes. It takes a lot longer than it would take for prokaryotes. So that makes life a lot easier for the prokaryotes in terms of cell division, but on the other hand, the DNA is not as nicely protected. So here we have a diagram of a nucleus, nice and big. You can see in red this thing here. That's the Nucleolus, the little nucleus. That's the site of ribosomal RNA synthesis. And you see these little pore structures right here. Those are the nuclear pores, because big stuff needs to go in and out of the nucleus. So we need to send messenger RNA out of the nucleus, and for that we need to have a special pore structure to allow that. Also, proteins need to come inside here, like transcription factors, so we need to have a larger pore structure. And here you have a transmission electron micrograph that shows a nucleus and the nucleolus, this region right here. And then moving on to the endomembrane system. First thing outside of the nucleus is always the endoplasmic reticulum, abbreviated ER. for endoplasmic reticulum. There are two types. The rough ER is called rough because it has ribosomes sitting on top of it, and you can see that all these little dots that are sitting on top of this endomembrane system, or here the little dots that you see all over that. Those are ribosomes sitting on the ER and giving it this rough appearance. Smooth ER does not have any ribosomes, and it's usually involved with lipid synthesis or lipid metabolism. Sometimes it works as a calcium storage place as well. And here, nice and big. You have the nucleus again with the nuclear pores just outside. We have the endoplasmic reticulum in green, and here we have some rough ER, these dots. So you can see the ribosome sitting on top. Out here in blue, that would be smooth ER without the ribosomes. No ribosomes on that one. Moving on to the Golgi complex. That would be sort of the next stop in the process of protein synthesis. And the Golgi apparatus is also an endomembrane system that sort of packages up protein products in little vesicles and transports them. So the most important part of the purpose of the Golgi apparatus is sort of the packaging and distribution center of the cell. So we have we getting the almost finished proteins from the endoplasmic reticulum. They hand it over to the Golgi complex, and then the Golgi will do final modifications and then package these proteins up in these secretory vesicles and then either ship them to the plasma membrane or ship them within the cell wherever they need to go. So here we have the Golgi complex. You see it looks like a stack of pancakes almost, and it's kind of closer to the border of the cell. So we have the nucleus right here, then we have the ER, and then here the Golgi apparatus sort of in gold or red right here. And you can see how it's another endomembrane system. These little vesicles, they arrive from the ER and then they get processed through the layers of the Golgi complex, and in the end we're going to pinch off these secretory vesicles that then contain a finished protein product, typically, that then gets shipped to the border of the cell or within the cell. And here, very important, another organelle that is very important for eukaryotic cells, the mitochondria. That's the power plant of the cell. So they are involved in cellular respiration, which means we are producing ATP, which is the energy currency of the cell. There we have another double membrane bound organelle. And so we already had a nucleus that has a double membrane enclosure. Now here in the mitochondria, you see the outer membrane is smooth. The inner membrane has these foldings that we call cristae. So these are the inner foldings. And that is where the electron transport chain takes place in the inner membrane of the mitochondria. Other organelles here include lysosomes. Those contain typically digestive enzymes that can be used to, let's say, a phagocytic cell that needs to digest a bacterium, or a cell wants to recycle a mitochondrium, a worn out organelle, then it will fuse a lysosome with the vesicle that contains, let's say, the bacterium, and then these digestive enzymes, they will pick that organelle or the bacterium apart. So digestive enzymes are in lysosomes. Vacuoles, they basically are little empty spaces. Think of them as the garage of the cell, and they can be used for storage. A lot of times you will find them in plant cells, where then the plant will start storing carbohydrates for extra energy. They also provide shape to the cell. But think of them kind of as the garage of the cell. And here in plant cells, the green ones here, we have chloroplasts, another double membrane bound organelle, and that's the location of photosynthesis. And so we have the green pigment chlorophyll inside of them. Gives the plants the green color, and then these flattened membranes inside here, these thylakoids. So you see these flattened membranes there, it's pointing it out right there, thylakoids, and that is the site of photosynthesis. And here, nice and big, we have this chloroplast right here. There you can see the thylakoids. They look like stacks of pennies, actually, green pennies, I guess. So you have these stacks, these membranes, and then outer membrane is smooth again of this chloroplast, and the inside we have these thylakoids. They form together these stacks of pennies that we call grana or granum if it's just one. And then here the two membranes. Again, we have three organelles that have double membrane enclosure. So that would be the chloroplast here, the mitochondria, and then the nucleus, of course. And then just to finish this off, so we have a couple more organelles that are present in many eukaryotic cells. That would be peroxisomes. They destroy toxic forms of oxygen, such as this peroxide right here, H2O2. And they can also oxidize fatty acids. And then in eukaryotic cells, we have centrosomes. That's necessary for cell division, for mitosis. So they form the mitotic spindle and have a central role in cell division and mitosis of the eukaryotes. Here a little bit, sort of, this is what we think eukaryotes came about. We think that the first cells, of course, were simple prokaryotic cells, and the first cells arose around 3.5 to 4 billion years ago, and then the first eukaryotes about 2.5 billion years ago. And so there's this endosymbiotic theory that I touched on earlier. Then that means that some bacteria, they were taking hold in the larger compartments of the eukaryotic cells, and in exchange they sort of cut a deal and said, hey, I'm going to help you with photosynthesis, or I'm going to help you with your energy metabolism. And so we think that the chloroplasts and the mitochondria of the eukaryotic cells, that they are sort of modified prokaryotes that took hold in the eukaryotic cells and sort of found some protection there. But they're doing also a job for the cell. So, and the reason why we think that is because the chloroplasts and the mitochondria, they have their own DNA, which very much resembles that of a prokaryote. So it's a circular chromosome, and the ribosomes in there also, they are the 70S ribosomes of a prokaryote. So we think that the chloroplasts, they were ingested photosynthetic bacteria that then got modified to become chloroplasts, and the mitochondria came about from aerobic bacteria that ended up being modified to become mitochondria. So here's the final slide for this pretty long lecture. We have the sort of the early cell. So this is sort of your first cell, and from there we have three branches ultimately splitting off. The bacteria, these are your eubacteria. Some of them are photosynthetic, that would be these green ones right here, and some of them are aerobic bacteria. We think, according to the endosymbiotic theory, the green ones, photosynthetic bacteria, they went into larger compartments of these eukaryotic cells to become chloroplasts, versus the aerobic bacteria, they went into larger compartments of eukaryotic cells to become then the mitochondria. And over here we have the archaea. They sort of formed their own branch ultimately, but early on they were kind of together with the eukaryotic cells that started developing. And here, of course, the big thing about the eukarya is that they did develop this membrane enclosed nucleus where the DNA is stored. So you can see over here the DNA first not enclosed, and then there's these double membrane enclosures formed, and here you have a true nucleus right here. And again, so then some of the prokaryotic cells decided to take hold in eukaryotes and decided to sort of have take on a modified function. And so the mitochondria and the chloroplasts of the eukaryotic cells are thought of as bacteria because they have 70S ribosomes and because they have a circular chromosome, so they have their own genetic material. Anyway, this is it for chapter 4. And so, of course, this lecture here, this segment was just about the eukaryotic cells, the profiles of eukaryotes, and to be continued.